Good afternoon, everyone. Thank you for joining GenScript webinar today. I see many attendees are still participating. Maybe we will give it one more minute for everyone to join in. Okay, so I will go start. Good afternoon, everyone. Thank you for joining GenScript webinar today. Today's talk is entitled Enhancing Precision Medicine, GenScript's Expansion into GMP-grade API Production and Neoantigen Peptide Synthesis. My name is Rachel Khan, and I'll be your host today. Today's webinar will run approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive and it works best when you involve. So please feel free to submit questions and comments for our speaker using the question chat box, and we will try to attend to your questions during Q&A session. This chat box is located at the bottom of the screen. Please note that this webinar will be recorded and be available for streaming on our GenScript webinar website. Now, allow me to introduce our speaker today, Dr. Guangrui Zhang, our Senior Project Manager in GMP Project Management Department. Dr. Zhang achieved his PhD in pharmacy from Tokyo University of Pharmacy and Life Science. Since his journey with GenScript from 2023, his work has been focused on to ensure peptide GMP project compliance and smooth delivery. Now I'd like to invite Dr. Zhang to start our webinar today. Dr. Zhang, please begin when you're ready.
Okay, thank you, Rachel. Would you mind close screen share? Okay, everyone, I will have double check. Does everyone can saw my screen?
Yes, we can see your screen.
Okay, so hello everyone. Thank you for giving me this opportunity to attend this activity. So today we'll be talking about enhancing precision medical, GenScript's explanation into GMP-grade API production and neoantigen peptide synthesis. So we will start with precision medical and introduce about OR effect in the field of peptide precision medical. So I'm going to divide my presentation into four parts. First is introduction and a peptide API, neoantigen peptide, and Q&A. So let's start with introduction. At the beginning of the introduction, we need to define that what is precision medical. So precision medical is proposed by Obama in 2015. So simply put, it is currently select and accurately apply the most suitable diagnostic and treatment method for each patient. And the proposal is first is minimized antagonic damage. It is request us as the raw material supplier to ensure that our product meet the quality standards required by our customer. And second is minimized medical expense. So it have to rely on our customer, which is a formulator. And third is maximum patient's benefit. So it have to require the effort of the end user, which is hospital or our patients. So the question goes to how we can to ensure the quality and safety of the product. So that we need the GMP standard system to ensure the quality of our product. So what is GMP and what can GMP guarantee product quality? So GMP, the full name is Good Manufacturing Practice. It is an independent management system that focuses on the quality and healthy safety of product and in the manufacturing process. So usually we used as the basic guideline for pharmaceutical production and quality management. It is applicable to key processes that affect the quality of finished product in the production of the raw material and even the entire process of pharmaceutical preparation production. And for the peptide raw material, GMP is most basic requirement for the IND filing from the pre-IND stage. And to be noticed that the GMP is not equal the sterile production. So they are nothing to do with whether a product is sterile. So what can GenScript provide to you guys? As we know, the entire life process of drug research and development, it will go through different stages such as discovery, preclinical development, and we goes IND filing, and we goes to the clinical trial. So GenScript can provide one-stop service for the whole process, and moreover, our QMS is phased and can adapt to the requirements of each stage. So our quality management system is established according to the ICH and FDA system-based compliance framework from the six major aspects as quality assurance, quality control, production, facility and equipment, material and the organization and the personnel. Our GMP facility located in Zhenjiang, China, with very modern and fancy building, and it has 30,000 square feet. And we have two GMP manufacturing line, one for the neoantigen and another one is for the peptide API. And at the same time, we also have the grade C manufacturing suite. And beside all of this, we've already launched the grade A isolator for the sterile peptide. From the last year July, we have delivered a total of 26 batches, support customer research from the pre-IND stage to the clinical trial stage, and successfully passed the audit by the 6 customers at home and abroad. At the same time, to help you better understand our GMP facility, we also took some photos, and you can visit our GMP factory. So first, on the left, in our GMP peptide production environment, we conduct adherence management of the environment. So this part is the normal area, and the blue area is the sea-level environment. And on the right is our peptide synthesizer. We have microwave reactor and reactors. As a result, we're able to deliver the large-scale peptide to our customer when in the very short turnaround time. And here I'll show our environment to arrange the purification, lyophilization, isolator, and in-process control for the GMP peptide. Also, we have so much modern facility has here, such as LC mass, HPLC to test the purity of the peptide, endotoxin analyzer, and the biosafety cabinets. All in all, we have the professional personnel and the professional equipment to provide you with the professional service. And beside all above, GenScript can also provide regularity support for your peptides if you plan to submit to the FDA or EMA and other agencies. And as we mentioned before that our GMP facility are designed to manufacturing line, here we show the different environment we prepare for them. For the API peptide, they are used as the drug substance in final product. Thus, the stereo is not mandatory. Therefore, the grade C environment is sufficient for the API. And for the neoantigen peptide, the produce of the synthesis and the cleavage are under the control at the no classes environment. And the purification and the lyophilization are in the grade C isolator. So sometimes application of the new antigen is being injected directly to the patient. Therefore, we have to need the grade A isolator to equip for the new antigen for the stereo. Next, I will introduce you one by one as the peptide API and the new antigen peptide. So let's start with the peptide API. For the peptide API, there are two characteristics of the peptide. API. One is general, non-personalized, and the definition sequence. And the other one is obtained by chemical synthesis, but not the fermentation. And there are two classes of the peptide API. One is the peptide used directly as drug, like hormones or general peptide vaccine. And the other is the peptide used for the conjugation, like antibody peptide conjugate. But if we contact the peptide API with the precision medical, the most famous peptide API is the semaglutide. So in the first half of 2023, the global sales of semaglutide ranked third among all drugs and first among of the peptide drugs. We can see all Novo Nordisk GLP-1 and the injection type semaglutide are exactly look like same, and they increased higher in the 2000, 2023. And for the GLP-1 agonists that are already on the market, they have removed the DPP4 enzyme recognition set. We can see it changed the alanine to the AIB to prevent the enzymatic degradation. Also, they added the fatty acid linker for the pro, for the prolonged serum half-life, and we can easily find that the GLP-1 has half-life for 1 to 2 minutes, but the semaglutide goes to the 165 hours. And for the tirzepatide, they are going to focus on the long-acting draw GLP-1 and the GIP receptor agonist. And for the chemical synthesis of the semaglutide, GenScript can provide two synthesis methods. So one is the full chemical synthesis, and two is the semi-synthesis. For the full chemical synthesis, we allow we allowed for very flexible amino acid modification and design, and we've already had some tests to show that the synthesized semaglutide is bioglycity active. And for the semi-synthesize, we use the 29 amino acid precursor biosynthesize, and we do the chemical modification. We added some fatty acid, and we added the histidine and AIB to ensure the short life for the semaglutide. And we have already succeeded the small scale batch and running to the pilot batch for the semi-synthesize. As for the GLP-1 agonist under the development, or we call the next generation GLP-1RA, and the future development detection is one, better therapy effect, and two is the low administration frequency. So for one way to achieve this is first is changing the main chain sequence, which we can do is the customized peptides. And for the second one is changing the side chain composition. We also can offer the customized peptide, and we also can offer the customized main chain peptides, which conjugate linker by customer themselves. You can add it whatever you want to the main chain peptide. And third is the conjugation with antibody. For the peptide conjugate with antibody, the most famous one is the AMG 133. So, and we can easily find that the advantage of AMG 133 like drugs first is it have the better therapeutic effect, and the second they have the lower administration frequency, and third, of course, they are easy to advise the painting programs. But there do have some pain points when we synthesize the GLP-1 peptide, like we have the low yield, low purity, we have difficult in the TFA removal, but we GenScript has completely opened up the synthesis process of the biomer acid GLP-1 analog peptides. So we can deliver the 1 gram this peptide at the 95% purity within 45 days. For the GLP-1 receptor agonistic, there do have some unmet needs, and for such as for the long-term runtime, we have the virus multi-channel peptide synthesizer, and also has many pre-synthesized fragments, which can further speed up the synthesis. And for the low success rate, we have the optimistic, the synthesized purification method of the GLP-1 peptide in the recent 1 year, and the overall success rate is over 95%. And for the internal question, the high price, we have accumulated experience in the GLP-1 like peptide production in recent 1 year, and we can provide the GLP-1 peptide in competitive price. So, as a brief summary, for the peptide API, we use GLP-1 as the example to show our capability. We also can provide the synthesis of long and complex peptide, including psychopeptide and peptide drug conjugate. And what we can provide to you is first experience. We have total synthesis of 1,000 GLP-1 like peptides, including the bioactive, immigrated, and the different bioamino acid GLP-1 peptide. Second is customized. We have the very flexible production form. We can provide region peptide, purified peptide, or peptide synthesized from the customer provided materials. And we also have the fast turnaround time and the low price. Finally, we can provide the R&D and IND support. We have the rich experience in our regulatory affairs team and the R&D team. I also want to share a case study with you. So we do have a customer from the pharmaceutical. Their stage is in the pre-IND stage. So what we do is we do the whole CMC study from the small scale to the IND filling batch, which is in the GMP grade. We do the production process development, analytical method development, analytical method validation, and the entire quality studies, including the purity profile study, reference standard structure characterizing, or and so on. Also, we do the stability study for the 1.5 year. And right now, we are signed in IND filing, which is now under the review by the FDA. Next, we go to the neoantigen peptide. So, first, what are the neoantigen peptide? The neoantigen peptide is a tumor-specific antigen only exists on the tumor cells, and they also have the personnel antigen and different between the patients. And for the neoantigen peptide, the application is first is the PCV, personalized cancer vaccine. Also, we have the personalized sodium tumor cell therapy. We co-culture with DC cells or T cells, something like this, and the injection to the patients and to target on the cancer. Also, we have the validation of vaccine effect, including mRNA neoantigen vaccine. So for the development of the neoantigen peptide, first, we do the in vivo tumor detection and we do the identification of the neoantigen peptides. And in this stage, our customer or partner of the customer will do the such as NGS to predictions the neoantigen peptide, or they will do, and after that, they will give us a kind of a whole sequence for the neoantigen peptide, like 100 or 1,000 neoantigen peptide, and we will synthesize out of them and give back to our customer. They will do the export of FACS to synthesize which is the target one to target the tumor they want to cure it for. After that, we will synthesize the neoantigen under the GMP facility or directly to synthesize the GMP grade neoantigen peptides. So in the whole link, the role of the GenScript is the synthesis of new antigen peptide. And then let's talk something boring for the mechanism of the new antigen. So the mechanism of the new antigen's action is the antigen presentation cells, such as DC cells, internalize the synthetic new antigen, which is over here, and he get into the cells to combine with the MHC. As we can see, the new antigen were present on the cell suffering with major histocompatibility complex, which is we call the MHC. So this new antigen combine with MHC and to combine with the receptor over the T cells, and the naive T cells will some differentiate into the killer T cells, and the killer T cells will move into the tumor tissue and attack the tumor cells that we can directly to cure the tumor. And then we will be recognized that there are four kinds of application of the neoantigen peptide. So first, peptides are directly injected and taken up by DC cells in the blood, and the peptides serve as a direct form of the treatment. And the second, peptide co-cultured with DC cells, injection peptide loading DC cells into patients, and and peptide as the key material, or we call the intermediate. And third is the are more likely mRNA vaccine. We turn the peptide sequence into mRNA encoding sequence, inject mRNA vaccine into patients. And fourth is engineering the TCR of the T cells to recognize the neoantigens. So we can easily class the neoantigen by therapy classification. We will separate them like directly injection, which we called peptide vaccine. This time, the peptide are used as the API, and it needs to be used with immune adjuvant and infuse it back into the human body in the form of the final preparation. And for cell therapy, they were separated with DC cell vaccine and the TCR-T therapies. So when we used peptide as the DC cells vaccine, the neoantigen are goes like the key raw materials, and the neoantigen will co-culture with DC cells and to loading the neoantigen back to the patients and target to tumor. And for the TCR-T therapies, the neoantigen are goes to like the immunogenicity screen and the validation to try to cure the patients. For the neoantigen peptide, we have lots of unmet needs, such as long-term runtime, and we found that most CRO company has limited requirement or lack automatic peptide synthesizer, and we GenScript have the hundred of automatic multi-chain peptide synthesizer, and for the GMP we have almost 20 multi-chain peptide synthesizer to provide a fast turnaround time to deliver door to door to our customers. And for the low success rate, so the neoantigen peptides are generally hydrophobic and thus difficult to be synthesized. And we have the new bioinformatic algorithm to predict the synthesis difficulty so that we can receive the peptide sequence and predict the difficulty of the synthesis or their hydrophilic, and we can give some suggestion to maybe you can have some exchange in the sequence to change some amino acid to increase their like pi or some hydrophobic. And for the large minimum quantity, we found most CRO company cannot provide milligram level GMP peptides, and Wiggin script has the specialisted neoantigen production line, which can which can only uh, which can only provide neoantigen by this line. and we can synthesize milligram grade GMP peptides. And for the high price, we can provide mixed QC for pooling peptide of the single patients. So for the new antigen, one patients maybe they will load it like 15 or 20 peptides in one time. If you want to do the QC items one by one, like you have to do the appearance, purity, impurity, TFA content, and so on, you will cost much more money than you think in the QC testing. So we can provide mixed QC, which for the pooling peptides, and to ensure in this one batch, peptides are all the peptides are under the quality standard that we can guarantee the quality of the neoantigen which we provide. Also, as a brief summary, for the neoantigen peptide, when you have to choose with GenScript, it is because first we have the experience. We've already supported 3 customers in the clinical trial neoantigen, and all of them are past IND in China. This number account is for more than half of the neoantigen peptide project that have passed IND in China. And then we can give you prediction. So we can predict for your sequence, and we can to prediction of the difficulty of the peptides, and we have the experience in synthesis and purification. And third, which is most important, is a stereo product. If you want to do the PCV, you have to directly inject the peptide solution to the patients. In that time, you have to need the sterile product. We can provide the milligram scale aliquot of neoantigen peptide and the sterilized peptide in the Q1, which we already launched. And finally, we can give you a quick turnaround time, which is under the 25 days. We can manufacturing GMP grade neoantigen peptide within 25 days. This is the manufacturing time. If you want to delivery, you can only plus like 3 or 4 days, and you can hold the neoantigen peptide door to door. And I also give a case study here. So one of our clients in the clinical phase 1, they have each patient's around 8 peptides, about the 20 amino acids. It stimulates DC cells ex vivo and then inject the DC cells back to the patients. What we do first, we prediction about the stability, and we have so much communication before we synthesize the peptides. We ask them to change some amino acid to increase the solubility of the peptide. And second, we development and transfer of the analytical method from our customers, which they already passed the IND filing. And the third, we provide phase 1 clinical trial productions. And right now we already provide like about 10 batches to our customers. So after talking about our API and the new antigens capabilities, there has to be a summary. As stated above, our capability to produce new antigen and APIs whose sequence lists purity, quantity, it is custom-made and can be adjusted. We usually synthesize neoantigen up to 35 amino acids, and the purity is crude to the 98%. Quantity is up to 150 milligram. And for the API, we can up to the 80 amino acids. Purity usually is over the 95%, or you can goes to the 98%. And the quantity is microgram to the kilogram. And they all can do the modification over 30 modifications. And for the new antigen peptide, as I said, we have the grade C background and plus grade A isolator to give you the serial peptides. And for the API, we have the grade C background. And for our capabilities, we can deliver to the 48 personnel the neoantigen sequence per patients per batch. And also, we can do the stereopeptide, and we can give you 500 peptides per month. And for the API, we can specialize in the synthesizing semiglutide long and complex peptides, including cyclic peptide and peptide drug conjugate. And we can deliver 400 gram per batch. And thank you for your listening. If you have any product needs, please contact our business personnel directly by the region.
Thank you, Dr. Zhang, for your presentation. Now we will start Q&A session. If there are any questions from the audience, please feel free to send in the chat box. It's located in the bottom of the screen. We'll wait for questions to come in. So for the first question, what is the production scale of a single batch?
Oh, we can provide the small scale production batch. It's up to. So as I showed in the final slide, we hold a second. We can provide for the API. We can provide 400 gram per batch. You can call it like we can this batch is for the GMP grade API. So if you want to start with the small batch, we also can do that. If just ask for the 400 under the 400 grams, and for the neoantigen peptide, we can provide 48 personnel neoantigen sequence per batch. uh, it have to be per patients because we have to ensure one patient's loading the whole this kind of neoantigen peptides so that we can do the pooling QC test for the patients.
Thank you. And the second one is there a minimum synthesis amount limit?
Yes, there do not have the minimum synthesized amount. So just like I said before, it all the things that you ask is under the 400 grams is okay for us. But if you directly ask for the like 0.1 milligram, we cannot do that.
Okay. And the other question is, if there is no CMC study, can we recommend corresponding quality standards to customers?
Yes, we can to give some uh, recommend of the quality standard for the API and the new antigen. But let's start with the API. So if you want to do some CMC study for the API, we can. If you want to do not have the CMC part with the API, we can directly give you a quality standard by our default. It will include appearance, TFA, residual solvent, and the purity. And if you want to add it some test items, we can also afford about that. And for the neoantigen peptide, we usually do not do the CMC study because for CMC study is specific for the each peptides. So as like I just said, if one patient's loading like 20 peptides, and we have to do the CMC for the each of them, we have to do 20 times CMC. So it's a huge money for everyone. Yes.
Okay, thank you. And what is the difference between parts of CMC and laboratory level research?
To be honest, it do have the uh, it do have the huge difference. So the CMC study is have the relationship with the production process, And if we use the different equipment and the different material, they will affect the peptide product. And for the quality studied part, which is the biggest part with the CMC part and the laboratory, even the laboratory's environment are almost the same as the uh, production environment, but we GenScript during the development, we have all the requirement and we have all the grade C environment to ensure the study is under the standard of the IND filing, and we will do the validation for we will do the PQ, IQ, OQ to all the requirement, and we will training our personnel. So we do not suggest we do we do not suggest our customers do the CMC part in the laboratory, and we suggest you come to the GenScript. Yes.
Thank you very much. I think that's every questions from the audience. So if you have any further questions, please feel free to email us or visit our website www.genscript.com, or you may also contact any of our local teams. So, and if there are no more audience questions from the audience, I think we can finish our webinar today. Additionally, I will share a link of today's webinar in the chat box, which you can rewatch today's webinar. Thank you for your informative talk today, Dr. Zhang, and thank you to all the attendees for joining us today. Hope you have a good rest of the day and goodbye. Thank you.